Hello, so today we're going to talk about mitosis. Remember, mitosis is the division of cells, somatic cells. Somatic cells are body cells, your normal skin, heart, lung, tissues, everything but your gametes, your reproductive cells. 4 phases: prophase, metaphase, anaphase, telophase, spells PMAT, okay? And this can last a few minutes to several days depending on the type of cells that we're talking about. The first phase is prophase. It lasts the longest, as you should have found in the lab we did. Here the chromosomes become visible. They condense, so they form those X's we're used to seeing. The centrioles migrate to opposite ends of the cell. Centrioles are those organelles that make the spindle fibers. Hey, guess what? They make the spindle fibers here. Okay? Microtubules that help separate the chromosomes are the spindle fibers. They attach to the centrioles and the chromosomes. Okay? They attach to the centromere of the chromosome. And, the nuclear envelope breaks down, and the nucleolus disappears. That's prophase. In metaphase, it's the second phase. It's very short. Basically, it's just one stop on the continuous chain of events that we have named, and this is where the chromosomes line up in the middle of the cell. So in prophase, they're all jumbled up. In metaphase, your cells line them up nicely in the middle. In anaphase, the third phase, the sister chromatids pull apart. Remember, the chromatids are the sides of the X's. They pull apart, and they become individual chromosomes. Originally, you had one duplicated chromosome, an X, and now you have 2 single chromosomes. Okay? And the chromosomes start moving towards the end. Anaphase technically stops when the chromosomes stop moving. Last phase is telophase. It's the reverse of prophase. Chromosomes begin to disperse, unravel. The nuclear envelope forms around each cluster of chromosomes to form nucleuses for both cells. Spindle fibers break apart, and the nucleolus starts to reappear. At this point, mitosis is done. Remember, mitosis is just the division of the nucleus, okay? But cell division itself is not done. Cell division has to go through one more step called cytokinesis. This is not technically a phase of mitosis. It happens right after / slightly during telophase. Telophase. And here's where the cytoplasm of the cell pinches together and divides. In animals, the cell actually pinches and pulls apart. Makes sense, there's no cell wall. In plants, there's cell walls, so we have to form what's called a cell plate, a division in the center of the cell that separates the 2 new cells and actually forms a new cell wall. Here's that in pictures. Okay? And you have interphase, the cell cycle. Interphase, G1, growth, normal cell, doing what it needs to do. S phase, it's going to duplicate. Okay? And then they didn't put G2 on here in a picture. They just put a little line that says, hey, it's in G2, it's preparing for cell division. Okay? Then you have prophase, chromosomes jumbled up but condensed, centrioles forming, nuclear envelope breaking down. Metaphase, chromosomes lined up. Anaphase, chromosomes pulled apart. Telophase, starting to unravel, nuclear envelope forming, no spindle fibers. And the start of cytokinesis. Here at cytokinesis, you have 2 new complete cells, and each cell will then go and make its own cycle. All right. Next week we'll talk, got one more thing. Here's what I was talking about with the plant versus animal. In the animal, we have what's called a cleavage furrow, where the cells pinch together. In a plant, we have what's called a cell plate, where it actually forms a barrier between the 2 cells. All right, next slide set, we'll talk about the control for the cell cycle.